that Josh gave and talk about something a little more cognitive, auditory social learning. And I think so social learning is a catch-all. There's all kinds of different types. And so the definition that I'm going to be using today is the acquisition of a new skill that's facilitated by experiencing conspecific, performing a well-defined behavior. And some people, it can be a behavior that the species would have performed anyways, and other people restricted to something that the species wouldn't have ever performed, so it's entirely conveyed by the demonstrator. So certainly musicianship or learning sports or even speech and language learning all fall under this category. One of the coolest examples is songbird learning. This is the adult father tutoring its juvenile son. And it's also a great, it presents one of the most interesting or most compelling examples showing the impact of social tutoring. So in this experiment, which was done in John Sakata's laboratory, an adult father tutored a juvenile, and at the very same time, every single vocalization produced by the adult father was passed into a nearby chamber of another juvenile of the exact same age. So this individual was yoked to the social tutoring, but only experienced the sound. So you can really separate sort of the incidental learning from experiencing sound from this sort of social attribute. There was also a controlled bird that had no tutoring. So here's the cool result. The social tutored bird had a much higher song similarity to its adult tutor than the yoked passive exposed. And so I'd say the difference between these two lines, that's social learning. That's sort of an effective social learning. But I'd also point out that there's some learning simply from being exposed to these acoustic elements. It actually works pretty well. And if you do it for a long time, songbirds can learn to mimic that song well. So the difference between the yoked passive exposure and the no tutor would be something like statistical learning or incidental learning of some sort. So the 2 points that I want to make is that social learning is involved in a lot of the natural learning that we do in many modalities, including the auditory system, but it's particularly profound during development. So I say it's extraordinarily important for children. From very early age, from infancy all the way through late childhood or adolescence. And the other interesting thing that goes along with it, it's involved in usually creating a skill. It's not involved in declarative learning and memory, where you're simply learning facts. It's involved in getting better at something. So practice is involved. So that'll ultimately be the message at the end of the talk. It's intermixed with a whole bunch of other forms of learning. And I think it's important to recognize that for humans and animals, I'll give you 2 examples, skill learning actually emerges gradually. So infants are great. We saw that already. In some ways, they're better than adults at certain sorts of acquisition. But it turns out that infants aren't in sort of an optimal sensory state or motor state to take advantage of everything they're learning at the time. And so one really cool example of this is an experiment from Bev Wright's laboratory in which they brought in adults, college-age students, and they brought in young kids, 11 or 14-year-olds, and they just asked them to practice on a very, very simple auditory task. And the auditory task was, which silent gap is longer? So first one, you could clearly hear the second one was longer. And so what they did is bring in people, again, at 2 different age groups, 3 different age groups, and they chose individuals that had the same initial thresholds. So individuals that could tell these 2 gaps apart down to about 20 milliseconds. And then they said, let's bring you back every day, give you a range of gaps and see how good you can get. And so the adults came in, they got 50% better. And then the way they brought 11-year-olds in and 14-year-olds in, they did something like this. They'd bounce around, even got a little worse. So these are old kids, right? They're driving tractors in Ohio. They're very sophisticated. They know what hypotheses are. But it really is the case that, that certain sorts of learning take a very long time to emerge. So I want to sort of hook this back to social learning. Social learning is extremely important. It facilitates, I think, skill learning, and it's particularly profound as we form and retain tight social interactions, usually with parents, sibs, and peers. So we actually did the same thing. We did use a different task, but we wanted to find out whether there was any sort of neural analog of this sort of plasticity, skill learning. So we used a different task than Bev Wright's group. We used amplitude modulation depth detection. So it sounds like this. Super easy. Much harder. And so we work on rodents, gerbils, and we trained them to do a task in which they had to release a lick spout that they were obtaining water from whenever they heard a warn trial. That was the amplitude modulated stimulus. If they did, it was a hit. And there were a number of safe trials interspersed. If they released the water spout, it was a false alarm. And we measured performance with D prime, Z scored hits versus minus Z scored false alarms. So when we did that, this is what we found for our adult animals versus our adolescent animals. It's an individual adult animal and then a whole bunch averaged together. I've sort of protected you from the details of the data here because I want to get across the concept rather than precisely what the difference was. And this was an individual adolescent and a whole bunch of adolescents. Yep. Yep. Yeah, so the graph. Okay, so the animals are performing the task that I showed you in which they release a lick spout when they hear an amplitude modulated sound. We make the depth of that sound of variable salience, and we're trying to find out the smallest depth, the smallest modulation in amplitude that those animals can detect, okay? And that would be their behavioral threshold. And so when animals come in, the first day, we show them how to do the procedure by using an extremely salient amplitude modulated signal, 100% modulation. And then we introduce more difficult values. So let's just look at this adolescent up here, this single animal. So it performed at this value for day 1 of training, day 2 of training, and day 3 of training. It actually got a little worse on day 4 of training, got a little better in day 5, worse again in day 6, jumped down in day 7 to be significantly better and stayed about there. Down is better. Yeah, so I'm sorry. So I made these gray so they wouldn't stand out, but good is better and poor is better. You couldn't see it. I'm sorry about that. Does that make more sense? Okay. More information? Okay, good. So basically, this sort of behavioral difference in animals that are really, they would live on their own in the wild, but they're adolescent. They're still in the middle of their hormonal surge. They're simply not learning as well as the adults. So we took another group of animals, and again, I'm sort of protecting you from the details of the experiment. We implanted electrodes into auditory cortex, and we record from animals wirelessly. So we can stream the data from individual nerve cells while they're training on the task and while they're learning. And when we did this, we found that individual auditory cortex neurons, I'm showing you an average, actually get better at discriminating between these amplitude modulated signals. Individual auditory neurons come to have a better amplitude modulation depth threshold during the course of 7 days of training. All of this is the work of Melissa Karras, who's now an assistant professor at University of Maryland. And the adolescent neurons, just like the adolescent animals themselves, actually improved relatively little. When we dug into the details, we found out that one of the reasons for that is they were more variable from trial to trial. That is, the number of action potentials produced from trial to trial was more highly variable than those of adults. So from a signal detection theory framework, the spread of activity was broader. In absolute terms, yes. The actual starting value might be different for the auditory cortex than the behavior.
So what I'm getting at is, are the individual neurons like on par with behavior?
The delta value is. The delta value is perfectly on par with behavior. However, any individual neuron might have a poorer amplitude modulation depth threshold either at the beginning or at the end. A population analysis like using an SVM or something, would definitely say that the population we recorded from could clearly explain that animal's behavioral threshold on that particular day of testing. But the individual neurons are kind of all over the place. Okay. Any other questions? Okay. So now I'm going to segue sort of into the social part of the talk. We study Mongolian gerbils. I haven't really introduced you to the species yet. Most people who work in wet labs use rats or mice. We use Mongolian gerbils for a couple of reasons. One is they're very social species. Adults pair bond, like prairie voles. They live as multi-generational families. So sibs stick around and their sexual maturation is suppressed for a generation, such that adolescents actually alloparent or help take care of their younger siblings. And they have a large number of vocalizations, much larger than mice or rats. And they have things like alarm calls to tell their friends of potential danger, and things like antiphonal calls. So this is something like turn taking. I call, you respond, I respond. And I'm just going to briefly show you a little bit of that data because I think it's relevant to the idea that these animals are using auditory information, natural auditory information in a social context. So this work was done by Ralph Peterson, and it's a collaboration between our lab and Alex Williams' lab and David Schneider's lab. And a lot of work was done by a rotating grad student, Violet Ivan. So we recorded a large group of vocalizations by monitoring families for weeks at a time. We have something like 500,000 vocalizations now, and we've pulled them out and we've presented them back to animals. There, that one you can hear. So this is actually a real, I downsampled it so you can hear it, but this is sort of a stream of vocalizations that the animals are producing when they're living together as a family. Don't know who does each one yet. That's actually the problem that Alex Williams, David Schneider, and I are trying to solve with deep neural networks that sort of sample from the environment and try to attribute vocalizations to individual animals in a complex setting. However, Ralph found that one of these vocalizations, when presented, produced a callback response. So it'll actually get lower. Respond for a very long time, and it's not a one-off. Most naturalistic natural behaviors tend to habituate quickly. This one doesn't. So, yes.
Sound waves of two animals or one animal?
So the playback is us. So it's a speaker that's above the cage. This whole response, the cyan line, that's all the animal that we're monitoring at the time. We don't have yet, I think, what you're alluding to, which is two live animals, one of which is calling, the other responds, and so forth. So we're setting that up now. Okay. So this is sort of a summary of the type of acoustic information that's traded. We play back a vocalization. It's ultrasonic, and this is a heat map of the response on a single trial of a single gerbil with sort of brighter colors, meaning more vocalizations per second. And this shows 25 trials taken sort of sequentially, one after another, with about 30-second intervals in one day. And so the animal's extremely reliable at responding after the playback starts. And if it's really chatty, that it continues to vocalize after the end of what I've shown you here, such that it's still vocalizing when the playback comes along, then it shuts up. So it's kind of a turn-taking type of sequence. And it's also present, we've now done this across a whole bunch of families. All of these animals are adolescents, in the middle of adolescence. It seems to be very profound during adolescence. All families do this. Some families are different than others. This particular family was quieter, although the recordings were made at the same age. So there may be sort of vocalizations that are dependent on position within the family, or it may be, entirely arbitrary. Yes.
Has been removed temporarily from the family in order
Yes.
to experiment.
Yes. Yeah. So, and one of the problems, we suspect in these weeks-long recordings we took, I presented you a few vocalizations, they're vocalizing all the time. They vocalize at an average rate of between 300 to 500 as a family, 300 to 500 vocalizations per hour. So they make a lot of vocalizations. We're recording them. We don't know who's doing it. So if we see a lot, we don't know whether there's one of these antiphonal responses going on. So now I'm finally going to transition into just a bit of social learning. How much time is left? Okay, great. And I want to go through sort of the basics, sort of circling back to the idea that different forms of learning are involved, and social learning being one of them. Yep. Yeah. Yes. Yeah, I should have mentioned this. Another reason we use gerbils is their hearing range is overlaps with ours almost completely. In addition, they have sort of the ultrasonic range that other small mammals have, like bats and mice. They vocalize both in the sonic range, the range we can hear, and in the ultrasonic range, the range that we can't hear. Say again? Yes. So we can record everything, and we have speakers that are good enough to play back everything. We know a lot about this one vocalization now, a lot or enough from research about another one, the one about warning friends. Those are both in the range that we can't hear. However, they do produce a huge number of vocalizations we can hear very easily when they're grouped together in a huddle, like the family's huddling together. That's something we noticed. I can't tell you if it's causative or anything like that. So 3 forms of learning I'm going to walk through. One is let's find out how well an animal can perform a task when it gets no information at all prior to attempting to perform it. So the task in this case is going to be super simple. It just has to differentiate one sound from another. The animal initiates the trial by poking its nose. It hears a relatively high rate amplitude modulated sound. I won't play it for you. And if it goes to a food tray, that's a hit and it gets a food reward. And on other trials, it hears a slower amplitude modulated sound of the sort I did play for you before. That's a no-go trial. And if the animal goes to a food tray to try to seek a reward in those trials, we score it as a false alarm. We turn out the house lights as a minor form of punishment. So let's just find out how well animals can do without anything, no experience at all. We put them in this environment and we say, we really pretty much say, figure it out. So the way to read this graph is that the x-axis is the day of what I've called exploration or practice. Exploration before they know anything, practice once they're starting to figure it out. And the y-axis is d prime, how well they were able to discriminate between these two amplitude modulated sounds. And what we found was that animals, on average, could kind of get this task after about 14 days. So 4 of the animals that we tested did. That's their average. 2 of the 6 animals didn't reach our criterion d prime, so they're fails at the end of that period of time. But that actually means that animals figure out for themselves. We've known that for decades, right? Animals that explore can find things. The second thing, and I think this might be a little bit more interesting, is that we wanted to run sort of a control for social experience in which the animals did experience the task cues that were ultimately going to be used by a demonstrator and by them. So here we placed them on one side of the cage. There was an opaque divider because we're studying auditory social learning, so we wanted to eliminate any visual cues. And we presented the task cues, meaning the 2 amplitude modulation rates. And we also triggered the release of food into the food tray because that actually makes a noise. And we yoked these animals to a set of stimuli that did occur under conditions when a demonstrator was performing. So just like the songbirds, they were getting exposed to sort of a real life sort of timing of all of these sensory attributes. after 5 days of this, we put them on the other side and said, how do you do? Okay, so read the graph the same way, day of exploration practice. Animals with no experience got it at about 14 days if they got it at all. And the animals that had access to task cues, on average, there was a lot of variance. Some animals got it really quickly, some took a long time, but they ended up taking about 8 days. So this is a big effect. They actually got it 6 days earlier on average than no information at all. And we attribute that to things like latent learning. So animals we walk around the world acquiring information all day long. This is sort of dates back to Tolman's experiments on maze exposure and maze learning. It's not a surprise, but it's a really important baseline if one is going to say that there's an effect of social learning. So finally, social learning. In this case, the naive observers on the same side, one side of the testing chamber. This time there's a live animal performing the task. So I'll just show you one trial of that. This is the demonstrator over here. He's going to trigger a trial in a second. There we go. He goes and gets it, and so forth. This guy runs over, and then they run back together. And when we watched them, it seemed like one of the social cues that the observer's picking up on is probably a localization cue. It kind of figures out where its friend is in space and follows it around from time to time.
Did you say that, I thought you said that they were on different sides of the screen?
Yes.
They couldn't get to each other.
They couldn't get to each other. So we've run it different ways. This particular way in the data I'm showing you, there was a plastic opaque divider between these two animals such that they couldn't touch each other or see each other. They could smell each other, they could hear each other, and I'd leave open the possibility, since they're sharing a floor, that they could sort of sense the vibration of each other. Those were the cues that were available. So here's the effect of social learning after going through what we originally thought of as controls, but now I think of as 3 different experiments. Social learning, the animals got it on average a little between 4 and 5 days. So the effect of social learning is about a 3 to 4 day improvement in what the animals already could have done simply by being exposed to task cues. And then the final piece of data, I think I'm going to just skip by this. Turns out vocalization is one of their cues, but I want to show you one last piece of data, which is the nervous system data. So we recorded from them, just as I showed you before, record from observers, and we compared the responses of single auditory cortex neurons, both at the beginning of social exposure and at the end. And what we were interested in here was, are there any changes to the neuron coding properties to the task cues that they were experiencing? And I'm not going to talk about the recordings that happened afterwards. They're also interesting. So here's the core finding at the neural level. Individual neurons, these are 91 neurons that we're relatively certain we recorded from for the entire 5 days, tended to improve their discriminability. That is, their D prime at being able to discriminate the 2 task cues got better when we averaged all neurons that we had, all responses that we had from the first 2 days of social exposure to the final 2 days. And basically 2/3 improved and 1/3 got worse or didn't change at all. And the task cue animals also had a bit of a change, but it was in the opposite direction. Some neurons improved their discriminability, but it was sort of a flaw. It was sort of a reversal. So 2/3 got worse or didn't improve at all or showed no change, and only 1/3 improved. So the neural finding in this case is somewhat in line with the behavioral finding, and we think largely explains it. So I think I'm going to stop here to see if there's any other questions. Yep. Josh.
So, yeah, I have a bunch of questions, as you might imagine. One that might be fun to think about is what about the social exposure drives this, right? I mean, you could imagine that the animal's just like perked up, when there's another animal next to it, right?
Yeah. Well, let me give you one answer from the literature, which is kind of the perk up. So the same group whose data I showed you in Songbird, vocal learning early on, they measured the activity in 3 separate neuromodulatory centers. The locus coeruleus, the VTA, so locus coeruleus, norepinephrine, VTA, dopamine, and I can't remember what the third one, maybe it might have been basal or something like that. The big change that they saw was activity in the locus coeruleus got much higher in the way they measured it, only in the social learning case. So that's kind of the arousal signal. That's a perk up signal. Ours would be data that I didn't show you, which basically says that the demonstrator vocalizes right around the time it nose pokes. And if we look at when they all occur for all of our demonstrators, it really piles up around this time. And if we go one step further, I'm sorry, and say, so how does demonstrator vocalizations relate to how well its friend is ultimately going to perform? The observers that had the chattiest demonstrators learned in the shortest period of time. So I'd say 2 cues to answer your question. I'm sure the localization cue is a big one. We haven't tested it. And then the other one is, and this might be purely arousal or it might be a timestamp, but vocalizations I'd come back to as they are a social cue. They're bound to elicit some kind of response, arousal or otherwise.
Okay.
Yeah.
Just on that one quick follow-up, do you find much more of these vocalizations when they're paired with somebody else, or do they do it also if they're just by themselves?
No, it seems like when they're paired. So when they're doing it on their own, there's very little vocalization. So if the demonstrator just goes in for the day alone, almost nothing. Yeah. Yes.
So suppose the demonstrator had the reversal of the actual cue. How would that affect the
demonstrator?
Speaking to the point about what the social effect is. So suppose you're hearing a bunch of vocalizations, but actually what the demonstrator is doing is responding to the opposite cue to the one that you're going to teach the gerbil.
Okay, so would this be the design would be that when the observer went over there, the cues, you would reverse the cues. That's right. Yeah, it's a great experiment. We haven't done it, and so I just have to say I don't know, but it's really a great experiment. We should do it.
Yeah, I mean the thought is that would be a way to discriminate between, is it just I'm social, I'm happy, I'm better at learning, or is it some information that you got from actually listening to the pattern of the vocals?
I think that's the best experiment. A poor one that we did do was we had an animal listening to its friend performing the task with light flashes. So we had, the animal was doing a task based on a go and no-go cue. The observer had no access to it. That observer did end up learning the task faster than it otherwise would have without any cues at all. So there's some advantage. That's my localization thing. I think it's listening to its friend, going back and forth.
Right.
But your experiment's better. Yeah.
I have 2 questions. One is this morning, we actually had little groups, in our group, where some people are here. We were discussing different modalities, like vision and motor control and some behavioral data from Allison. Whereas we were saying how in different modalities, when babies are born, there's a lot of noise in the system. And through the developmental kind of curriculum, we kind of do the simulated annealing, maybe, of getting to some local minimum that is highly performant as an adult. And we didn't know anything about the audio domain. And so as a developmental audio person, I was just going to ask you something, and maybe your juvenile results kind of show that. Is that something that also happens in the audio?
So are you talking about perceptual attributes or something more cognitive like attention?
Perceptual.
Okay, so let's see. There's so here it is. I did a review a long time ago with Sarah Woolley, in which we actually just walked through the literature. This is psychophysics experiments from humans. Human infants, kids come into the lab. It's earphones, soundproof booths, so there's nothing naturalistic about it. But we walked through every study that had sort of been done on infants, and we asked, when did you reach adult, essentially adult level thresholds? Does that come closer to what you're thinking about?
Sure.
Yeah. So the take-home message was it's slow and asynchronous. So some things like frequency resolution, this is our ability to hear out something in the presence of another sound, actually develops really early. This is Lynn Werner's work. A lot of this is Lynn Werner's work. Gap detection, just the ability to hear the shortest possible period of silence, which we use for phoneme discrimination, doesn't really mature until 5, 6 years of age. Something like sound localization depends what you're asking kids to do and how hard the task is, but they get pretty good at it around 6 years of age. Duration discrimination, more like 6, 7, 8. Frequency modulation detection, and so forth. And the longest one that I know of is amplitude modulation depth detection. So, I have no idea. I don't have an explanation for it. I don't know why it would be. I'd also say it's a level cue. Level cues are interesting. If you just ask people to sort of walk into, Josh is probably, I'll defer to him if he has a different answer on this, but if you ask people to walk into the lab and you test them on intraoral level difference, how small of a difference can you test between your two ears? It's like we walk in all over the map. However, if we train on the task, we all get, much better. But it's a late developing cue.
It's super late. There's all this is super late. This is like,
It's-
5, 6 years.
Well, it varies. I mean, there's others that aren't up here, but I would say, from my point of view, again, infants are amazing, and Lynn Werner has really devoted her career to showing just how amazing infants are at doing all kinds of auditory tasks. So I could fill in the bottom of this with her career, but there are a bunch of other studies that show late developing adult thresholds.
So I'm going to do one more. So I'm going to do one more. Because you're not coming, so at the end of the month, we're going to have a social part of our workshop. And I know you can come to that, but because you're here now, and you showed this interesting stuff, I was wondering, it's kind of like for you and for Allison together, because Allison has this theory about caregiving and how that's important for the development of human intelligence, caregiving for younger specimens, children. And from what you're saying, I'm wondering if it has to do really with learning and social learning as you're younger. Maybe that's, I don't know, this kind of like
I mean, you're talking about the question that we most want to address in the laboratory, by being able to monitor animals in their home environment continuously, not disturb them, keep track of everybody, everything they do, every vocalization they make.
Is there any evidence that that the animals learn more from the alloparents?
No, we don't know. We really don't anything about sort of a natural version of anything I've talked about. The closest that we came was like, 2 sides of a cage. And that's truly the goal. The goal is that's really what we're doing with Alex Williams' lab and David Schneider, is setting up a large environment with an underground burrow, because these animals live underground, and none of us have ever given them access to the normal way of growing up in the dark. So they do all of this stuff, all this initial vocalization in the dark. There is no vision for these animals while they're initially. And we don't know.
You should make friends.
Yeah.
Okay, so we have a good opportunity for that. We have a break now. So let's chat more over the break. Let's thank Dan. Why don't we say 3:20, we'll come back. So about 20, 25 minutes. 3:20.